Okay, guys, this is a first for me, so bear with me. My children are going to come in, and I'm going to have to handle that and do all that stuff in between. So we will get through this lecture, and I hope you enjoy being able to push the replay and listening to it over if you need to and things like that. So, okay, here we go. Review of the cell structure and organelle function. So on Tuesday, we will have gone over everything that we're going to do for the year, and then we're going to do some of the beginning lecture. I'm trying to anticipate where we might get in our lecture to have you prepared for your test. So we will start on page 13, the review of the cell structure and the organelle function. So we're going to skip the tissue in this section because we have a whole chapter on that next, going over all the tissue. We're going to jump to the cell and to its organelles. So they told us that we would be going over gross anatomy, that would be our focus for the year, but they get really detailed here. So good to have a really basic understanding of the cell. You went over this in biology. We're going to do a quick refresher of it here. Again, I told you on Tuesday, if you don't make fun of my drawings, I will not make fun of yours. So you're going to get some really good drawings today. All right, figure 1.7, you will need to know that for your test. So make sure that you are able to identify those pieces and look at that. So make sure you are familiar with it. And as we kind of learn, it's going to ask you some questions about it. You're not going to have to label it or anything like that, but it's going to ask you some questions about some of these items that we learned about today. And I'll point those out as we get to them. Okay, so not every cell will have all of these features, but some cells will have some of these depending on the function of the cell. And we're going to learn about that the rest of the year. What the cell's function is depends on what types of these things that they have. So the tissues are composed of cells, and cells are composed of organelles. So it's good to know the major organelles and their major functions. So I'm gonna get my little picture ready so that I can draw. The first thing that we are going to learn about is the plasma membrane. And the plasma membrane is the boundary of the cell that holds it together. It controls the in and outs of the substances. Also, lots of receptors there to help it do its job. So we've got our lovely little plasma membrane here. The next three, the nucleus, the chromatin, and the chromosomes, are the control center. So the nucleus, the whole thing is called the nucleus. So we have let me get a different color marker here. We have our nucleus, and the nucleus is wrapped in a double membrane, very porous. It also contains our DNA inside the nucleus. DNA stands for deoxyribonucleic acid. It is the control center of the cell because it has the DNA, so it's the control center. Next, we have the chromatin. That's what makes up the nucleus. It has the DNA there. The chromatin contains the chromosomes. Chromosomes are the structure of the DNA. It's the coil. It's the thing that gives you that pretty little XX coil that you get to see on lots of different things when they're talking about DNA. So that is the chromosomes are what make that up. So we have the plasma membrane and we have our nucleus. Then the cytoplasm here, the cytoplasm is between the nucleus and the cell membrane, and it contains, they have it, it is not labeled in your picture here, but the cytoplasm between the nucleus and the cell membrane, and it contains cystol. Cystol, again, cystol is not labeled on the picture, but cystol is a fluid containing all kinds of dissolved chemicals like ions and proteins and other molecules and all kinds of things like that. So that is a fluid inside the cell, in between, kind of holding all that stuff together. That's the fluid in there. Next, you have the ribosomes. This is labeled, and these things are the little dots. You can see tiny little dots all over the picture here. and those are the ribosomes, really, really tiny little dots. Ribosomes are like the kitchen of the cell. Okay, so proteins are produced here. They're the type of protein. The type of protein that is produced in the cell determines what type of cell it is and how the cell is going to function. Sometimes ribosomes are attached to an organelle, and if a ribosome is attached to the organelle, that is called an endoplasmic reticulum. So those of you who've had Latin before, right? Endo is within, and we're going to have lots of Latin words. If you've had it, it'll help you throughout the semester, helping break down these words and what they mean. So vocab should be easy for you. So endo is within. Plasmic is the cell cytoplasm. So within the cell cytoplasm, and reticulum is a network. So within the cell cytoplasm, a network, and it is specifically a network within the cell cytoplasm. So here you have two types of the endoplasmic reticulum, and these are pictured. You have a smooth endoplasmic reticulum. Let's see, let me get a picture drawn here for you. The smooth endoplasmic reticulum. Oh gosh, this is terrible drawing for me. Oh, I need to switch my colors too. Endoplasmic reticulum kind of comes off of the nucleus, and it has a little outer wrapping layer. So you have a smooth endoplasmic reticulum that kind of looks like some little silly string coming off of the cell nucleus. On page 14 you can see it there. And then you have the rough endoplasmic reticulum that kind of looks like a maze. Also, they have the ribosomes attached to them inside the maze. So I'm going to put some little dots in there so you can see that. So, terrible. Smooth endoplasmic reticulum, the silly string coming off the cell. Rough endoplasmic reticulum, it looks like the maze and has the ribosomes attached to it there. The smooth, so the two types, the smooth are tubes that are used for transport within the cell. The production of lipids and carbs take place here in the smooth endoplasmic reticulum. The rough endoplasmic reticulum, the maze-looking thing, is also intercellular transport. It's going to be transporting things inside the cell. It's rough because it has the ribosomes on it. You notice the smooth does not have any ribosomes. The rough has the little dots of the ribosomes. And again, you can see a much better picture in figure 1.7. So it is used for protein synthesis, also making the proteins and transporting the proteins. That is what the endoplasmic reticulum is for. All right, next, one of my favorite things we'll learn about all year is the Golgi apparatus. It's really cool, and it's in lots of different systems. The Golgi apparatus is the cell's packaging plant. It packages chemicals for different uses, mostly secretions. Saliva glands have lots of the Golgi apparatus here because they're going to secrete saliva. It will hold it until it needs to use it, and then it will secrete it whenever you need it. So Golgi apparatus is really, really cool. And let me get a picture of that here. It's also off kind of to the side. Oh, my purple's not wanting to work. Let's see if this purple works. Some little ribbon looking things. Nope. Purple said no go. Off to the side. Golgi apparatus, the packaging plant where it's going to package the stuff for you, hold it in the little sacks until it's ready to be released or to be used. So it's for secretions. It's going to hold the chemicals, mostly secretions for the use. Then you have secretory vesicle. The secretory vesicle, this are just tiny sacs, and I'm going to make them just little green bubbles here. So the secretory vesicles are, once the Golgi apparatus, these little bubbles here, once the Golgi apparatus packages the chemical for secretions, it puts it into the tiny little sacs and it pinches it off of the Golgi apparatus. So here it's packaging it. Once it gets it nice and packaged how it wants it to, it pinches off of the Golgi apparatus and it hangs out here, traveling through that membrane to the outside of the membrane where the chemical can be released. So it pinches it off into these secretory vesicles, okay, the little green things, and it is going to travel outside of the cell membrane where it can be used. It builds up into these little sacs until they're ready to be used again. Another really awesome feature of the cell is a lysosome. So cool. It's a kind of vesicle. Its main function is to break down lipids, proteins, polysaccharides, carbs, and nucleic acid. So in order for it to do its job, it must have a certain enzymes. The cool thing is, if these, let me get you a picture of a little lysosome. Okay, it's going to be kind of bright yellow in here. So the lysosome, if the enzymes are released to the cell, it can damage it and even kill it. And so cool. So have you ever heard that you can live only about 4 to 8 minutes without oxygen? It's because of these lysosomes. They can't hold it together, okay? They're gonna burst, they're gonna dump their lethal enzymes into the cell. It's gonna kill the neurons, and that's when it's bad. But it can also be for a good thing, right? So the lysosome being a bad thing. If you don't have enough oxygen for a certain amount of time, the lysosomes are going to lose it, can't hold it together, they rupture, they're going to kill the cell, and that is what causes the death there, because you're having cell death. But it can be a good thing for the lysosome to rupture, like when you have a diseased or a damaged cell, it's going to help those cells to die off faster. The lysosomes will rupture and it'll kill the cell, and that is the end of that bad or damaged cell. So white blood cells are full of lysosomes, full. They are going to help you get the infection out. So the white pus, like if you have some sort of bump, bite, sore, something like that, and it has white pus coming out of it, that white pus is the busted lysosomes from your white blood cells. So if you have some sort of thing, injury, bite, any of that, the white blood cells are going to rush to there to help fight infection. Well, once those cells are damaged or not any good, the lysosomes are going to rupture. That white pus there from the white blood cell is what is kind of coming out. So it kills the white blood cell. The lysosomes rupture and kill that white blood cell, but it's going to kill that foreign material there as well. Pretty cool. All right, next is the centrioles. These are neat looking little doodads, and they look like this, and they have these little finger things kind of coming off of them. So the centrioles here in our green. Centrioles are found in the centrosome, which is the center of the microtubule. Let me get a microtubule, are the little lines kind of growing off of it. So the centriole right in the center, the circle, the microtubules are those tiny little lines there. They're spiral strands of protein, and they form a rope-like structure. It influences the movement and the shape of the cell. The centrioles are really important in cell production. All right, next are the cilia. These are really cool as well. And cilia are on lots of things, and you're gonna really be amazed at how many things actually have this. Cilia are like tiny hairs that formed from an intricate arrangement of microtubules. You have them in the back of the nose, you have them in your throat, in your larger airways. They help move things upward to help you clear mucus and dust. So if you get the mucus or dust like in your nose or you breathe it in through your mouth and it goes into your lungs, those cilia or little hairs, they're going to help move it to the top so that you can move those dust particles or the mucus to help you get it out of your system, to help you cough it out. So the cilia are the little tiny hairs that are going to help you get rid of things like that. I just drew some little tiny hair looking things here for the cilia. Also, you have microfilaments. And let's see, different color. Microfilaments are on here. They also help with the movement. So I did a dark green microfilaments. This allows some of the cells to contract, okay? Like a muscle cell. It's gonna help it to contract in. And these are just right around the cell membrane to help it contract in. Mitochondria. This, you need to know what the mitochondria is. The mitochondria is the major site of ATP production. ATP is adenosine triphosphate. This is what gives us our energy. Okay, ATP is energy, adenosine triphosphate. Mitochondria is the power plant of the cell. It's the thing that is making the energy for the cell. It's also how the energy is stored. So the majority of the energy is produced here. It's how it's stored there. All of that. It is a biggie. You need to know the mitochondria. Let me get the mitochondria on here, and I need a new color. Let's see, mitochondria. There, I made it a big blue. Mitochondria. Okay, that is the thing that is going to be the power plant, stores all the ATP for energy, and all of that. So these are the major things of the cell. Again, remember mitochondria, know what some of them do. We're going to come over them again multiple times. The Golgi apparatus, the lysosomes, the cilia, all of those things we're going to talk about quite frequently. So try to remember what they are, but absolutely know what the mitochondria is. Okay, don't laugh at my lovely little picture. Okay, protein synthesis. We are now on page 17. So proteins are large molecules that are formed by the joining of amino acids. How they are formed together determines what that specific protein does. So some are catalysts, they speed up reactions. Others act as hormones. Some act as antibodies, which are going to fight infection. So tons of different proteins are involved in the life process. So you have 2 steps of protein synthesis. Synthesis means making, protein making. So 2 steps here. You have a transcription and translation. So transcription, this is the written representation of something. The translation is the process of rendering the meaning of one thing into something else. So DNA, it has 4 letters here. The A is adenine, T, thymine, C, cysteine, G, guanine. These 4 bases are used in groups of 2 to form words that make up genes. So A, T, and C and G. So a cell can transcribe a gene, it can write a word, then it can translate it. It can copy it into a complete protein. So it can write it, and then it can copy it into a complete protein. And figure 1.8 shows you how this happens on page 19. There's the little picture of the DNA that you guys will all recognize. So that shows you how that works. Okay, now cellular mitosis, figure 1.9. Again, this is just a brief review introduction. I'll tell you what you need to be familiar with. It's just kind of getting the basis going for the cell so that we can get on to the body. So cellular mitosis is cell reproduction. There are 4 broad steps. You have prophase, metaphase, anaphase, telophase. Know those 4 steps. Prophase, metaphase, anaphase, telophase. You went over this in biology. I'm sure you remember it. Interphase is the state when it's not undergoing reproduction. It's just the normal state for a living cell. Okay, when it's not in reproduction, it's in interphase. The reproduction phases are prophase, metaphase, anaphase, telophase. Know what those are. Okay. Prophase, there are 4 things happening here. The centrioles duplicate. You can follow along in your book in figure 1.9 as I go over these. The centrioles are duplicating to begin to form microtubules. They're moving toward the opposite ends of the cell so that the spindle is going to spread across the cell. Replicated DNA forms the chromosomes, which are going to move towards the center of the cell. which is the equatorial plane, and you can see that in the picture. Then metaphase, the spindle is going to attach to the chromosomes right at the point where it is replicated. The chromosome is attached to its partner. Now the spindle begins to pull, so it duplicates and originals are pulled apart. Anaphase, duplicates and originals separate and are pulled in opposite ends of the cell. So they're being pulled apart to opposite ends of the cell. Telophase is one set of chromatin on one side of the cell and another set on the other side. The plasma membrane is going to constrict in the middle, and it's going, so they're being pulled to both sides. It's going to constrict in the middle and pinch the cell in two, and it now becomes two. So the result is that you have two daughter cells that go back to interphase. They're not reproducing at that point in time. So back to interphase. You get the X shapes during the prophase and the metaphase. They exist because the chromosomes have been duplicated. If it hasn't been duplicated, you do not have that X shape you can see in the picture. Okay, 3 types of cells that cannot duplicate: mature neurons, mature skeletal muscle cells, and mature cardiac muscle cells. This is important, not necessarily for the test, but just for life, for you to realize that these things cannot ever duplicate. They can never make more if something has been damaged to them. So they don't have centrioles, so they cannot form that spindle. They cannot grow new ones. They cannot do that. So the centriole right here from the middle that helps hold its shape with the microtubules helping it form the shape and the function, it doesn't have any of those, so it cannot help form a new cell. So you cannot grow any of those new ones. Your mature neurons, your mature skeletal muscle cells, and mature cardiac muscle cells cannot make any of those new. Okay, I'm going to erase my beautiful cell because we are moving on to the plasma membrane. Okay, so the plasma membrane, page 21, the plasma membrane, it holds the cell together. It restricts what goes in and out. So again, we're talking about the plasma, the big circle thing on the outside of the cell, the plasma membrane holds it together. It restricts what goes in and out. It lets it lets the cell communicate with the environment around it. You can see a picture of figure 1.10, what the plasma membrane looks like. So I drew it as this for the cell, okay? But you can see in the picture on figure 1.10 that it's actually 2 layers here. So it has a phospholipid bilayer. So an outer layer of the membrane, it's got 2 fatty acids and a phosphate group. It's polar, which means it's water soluble on one side, and it's non-polar, lipid soluble on the other side. It's got 2 stems. So it's got, let me erase this again. And something smaller. Okay. So it's got the end, it's got 2 stems on it. So it's got the phosphate here, which is polar, which is water soluble. And it has the 2 stems, which are fatty acids. These are non-polar. So you've got your polar and your non-polar here, the 2 fatty acids and a phosphate group. So it's got your phosphate, which is the polar, and the fatty acid. So the bilayer is that it has 2 layers of this, okay? 2 layers. It has a top layer and it has a bottom layer, and it's just like this. They attach, you can see the much better picture in your book. Again, they attach like this, and they look like little jellyfish, rows and rows and rows of jellyfish. So it has a head and 2 tails, the water soluble or water loving head, and the tails, which will dissolve in oil. They're water hating. So because of this, it can reform if it gets disturbed. So the tails are attached to each other. The heads are attached to each other as well. So these things get nice and close like this, and they can easily really reorient themselves, and the heads are drawn to the water on the outside of the cell, so it can reform and draws it this way. But these oil-loving lipid-soluble tails are also drawn to each other, so they are pulling the cell this way as it's pulling the cell that way. So the inside and out is pulling to help keep the form of the cell together. All right, that lipid solubility, the lipid barrier between the two different water-based fluids. You have an intracellular fluid and an extracellular fluid. I thought I heard the baby. So water-soluble heads, but mainly this lipid-soluble barrier here. So inside you have these channel proteins. Let me make my picture a little bit better. So inside you have lots and lots of those things there that make up a whole row of the plasma membrane. But you have channel proteins, which are used for your larger molecules. It's a channel to let water and small molecules in and out, and it kind of looks like this. For the plasma membrane, it's going to be open like this. It's a channel protein. It's going to let those things get through the larger molecules and water and all of those things that need to go through the membrane. It's going to let those things in. And then you have glycoproteins. These are because glucose, so glyco means glucose, so glycoproteins is a protein with a carb chain attached to it. These markers are really just kind of allowing for recognition here. So glycoprotein, oh, I need a new color. And it's going to have a little attachment with a protein with a carb attached to it there. Then you have receptor proteins. These things are going to receive messages from other proteins. So the receptor proteins there are the things that are going to get the messages back and forth from the other things. Glycolipids, it's a lipid with an attached carb. You can see a picture of that. They have it green in the picture. Oh, I'm running out of colors. It just kind of goes through that little top layer, and it looks like this, okay? The glycolipid, it helps to anchor the proteins and provides some structural support for the plasma membrane. Then you have cholesterol that makes up about 1/3 of the lipid part of the membrane. Cholesterol gets a bad rap. You need cholesterol in your body. Of course, they like to go on and on and on about too much cholesterol, but you still do need cholesterol. 1/3 of the lipid part of the membrane is lipid-soluble nonpolar. So it's found by the tails, okay? Lipid protein down by the tails. Very, very important to the membrane, not a bad guy. It's used to help stabilize that membrane. It helps give it its firmness so that it doesn't fall apart after a while. So it helps the structure of that cell. All right. Next we have all right, the functions of the plasma membrane are, remember we talked about it, it holds the cell together. It provides the receptors so that the cell can sense its environment around it. So if you have type 1 diabetes, you can't make insulin. The insulin signals the plasma membrane to let the glucose enter the cells. If you have too much glucose in your blood system, the insulin will signal that plasma membrane so that the cells will take in all that extra glucose. Type 2 diabetes, receptors are not going to work correctly at all. Or if you have too few of them, the receptors are really important, so you need them. So the plasma membrane is going to have the receptors on it to tell you if you have too much water, or too much sodium, or too much glucose, or any of the things like that, whether you need to let it in or to let it out. So the receptors are very important. Also, Selective permeability. You need to know what that means for the test. So selective permeability is the ability to let certain materials in or out while restricting others. Really easy. It's selective permeability. Whether it's letting certain materials in or out, it's being selective, restricting others, letting other things in or out. So selective permeability, it has this lipid soluble nature. So nonpolar. It's going to get in very easily because the membrane is largely lipid. It lets in lots of fat, of course, because fats are lipids. So it has that lipid soluble nature. It wants to let those things in. Channel proteins that are going to allow small molecules, this thing right here. So it has the channel proteins that are allowing those small molecules such as water move in and out of the cell really easily. Larger molecules like proteins don't really move easily at all. You have the channel proteins can actually have a charge. They can have a positive or a negative charge, and they will only allow ions in that they are attracting. Remember, opposites attract. So if it's a positive channel protein, it's going to attract the negatives. If it's negative, it's going to attract the positive. Now, the next thing that it does is a little bit more complicated. It is called mediated transport. This is, I have a picture of it on figure 111, mediated transport. These are carrier proteins. They accept molecules with a particular shape or binding, okay? 3 conditions must be met for these carrier proteins. Specificity, specifically shaped protein. You can see it's got this Pac-Man looking thing in here, okay? Only something that matches exactly can fit into that carrier protein, okay? If it's a triangle shape, if it's a square shape, whatever it is, whatever shape it is, it will only allow that specific protein to go into it. So specificity, specifically shaped protein. Now competition. Similar shapes are going to compete over that spot. So if the rectangle thinks it might be able to slip into the square slot, it's going to try to do that. Also saturation. If it gets full, it gets full. It's going to limit the amount of molecules into a cell at one time. If the cell is full of that, it's going to turn it off, shut down. It's not going to allow anything else in. All right, the transport process. You have 2 kinds here. You need to understand how these transport processes work. You have passive transport, which is diffusion. Okay, it's just through diffusion. The movement of the ions or the molecules from an area of higher concentration to lower. They don't like being with the crowd. So you've got a crowded room, you can hardly move around, too much sodium. Our bodies always have too much sodium. So much sodium is in an area and it wants to go from the higher concentration to the lower. It's always trying to get out. That's just called diffusion. It's passive transport because it doesn't need any energy. It happens on its own. Diffusion happens on its own. Higher concentration to a lower concentration. Passive, it doesn't require energy. So passive transport. Then facilitated diffusion. This is a diffusion through that mediated transport through those carrier proteins. A higher concentration to a lower concentration through the carrier proteins. No energy is needed here. So those are the different types of diffusion, and facilitated diffusion are passive. No energy required. Now, active transport is the second type of transport, and active, the transport that requires energy, or ATP. Remember, adenosine triphosphate is energy. It requires that energy to move the molecules or the ions across the membrane. So a lower concentration of something, it's going to force it, it's going to take energy to move it to that higher concentration. It goes against the grain. It's having to work harder. You're swimming upriver here, okay? If you've got a really introverted person, you've got a crowded room, and you're really having to force them or use your energy to push them into that crowded room, right? Same thing here, active transport. Again, we'll use sodium. Too much sodium will kill a cell, but our bodies are always very high in sodium. It's a higher outside constantly. It's a lower inside the cell because too much sodium can kill that cell. So it's always a lower inside the cell, higher on the outside of the cell. And it's wanting to push it if it needs to get rid of it, out of the cell, because it might still be a lower concentration in the cell than outside of the cell, but it's got to get it out. It doesn't want to go back out, right? It's too crowded outside of the cell because it's a higher concentration on the outside. It's got to force it out, and it must expend energy to force it out. Active transport. This is called pumping. So the water is going against the gravity here. Pumping, the same thing. When you're pumping up water's going against gravity. Pumping here, same type of thing. You're having to use energy to make it go against what is natural, pushing it out. All right, 2 types of active transport where it requires energy to move it. You have endocytosis and exocytosis. We'll talk about this frequently as well, so make sure you understand what this is. It's got a picture of it in figure 112. You can see endocytosis on page 29. So the process where the large molecules are taken into the cell. So 2 parts here to this endocytosis. You have penocytosis. Know what active transport is. Make sure you understand that. Know what penocytosis is. This is cell drinking. The proteins are dissolved in a fluid, and it enters the cell. So it's cell drinking. Phagocytosis is cell eating. It ingests the particles rather than the fluid with the proteins. So it grabs it and it folds it inward into the cell, which you can see in figure 112. This is penocytosis. So that's an endocytosis, where it's taking it from outside of the cell into the cell. Now exocytosis is where it takes it from in the cell and it puts it out. So it's the transport of material from inside the cell to outside of the cell using vesicles, usually secretions. So the Golgi apparatus, remember we just talked about that. Figure 114 gives you a picture of this. So remember, we're having to use energy, ATP, because of the size of the molecule. It's not the higher or lower concentration here. It's because the molecule is too big. All right, and you can see that picture of the Golgi apparatus taking that secretion that it made. It held it into the little vesicle, and then once the body needed it, it pinched it off, and then it sent it outside of the cell. All right. Oh my gosh, we totally just finished all of that. I cannot even believe it. That's crazy. Okay, so that clearly means I was talking way too fast. So you'll probably have to rewatch it. I'm sorry. Okay, for your test, which we've talked about Tuesday, this is such a short chapter and it's such a review. The other chapters are much longer and much more difficult, and I want to get through this quickly. So test on Tuesday. Please email me or text me. You have my number on the schedule that I sent out. Any questions that you have, and I will get back to you very quickly. So make sure you know for your test the 7 levels of organization. You have the 4 types of tissue, just the 4 main types of tissue. Understand homeostasis, just have an understanding of what that is. What organ controls the ATP? I know you remember that. It makes it and it stores it. It's the powerhouse of the cell. Know what that is. Know the 4 phases of mitosis. Know what those are. And your powerhouse organs. Know what those are. Several questions on what we've gone over today. So fatty acids, proteins, carbs, and how they're being transported through the cell. So the transport systems. Let's see what else. Okay, so you've got some definitions and the 7 levels of organization, some different types of tissue, identifying the types of tissue, and positive, negative feedback system, being able to recognize what that is, what the control center is, what the effector is, the small polar molecules, how remember again the transport systems affected in the cell, how the small molecules go in, how the larger molecules are going in, and how the very large molecules are going in. Know how that happens. Also, know about what we talked about with the sodium being transported in and out, like what has to happen because of that active transport. Make sure you know about endocytosis and exocytosis. Understand what those are. Okay, and I think that's it. Again, please remember to text me or email me with any questions, and I will see you guys Tuesday. We will do a review before the test, and then we will take the test in class. As soon as the test is, that's it. We just take the test. We don't start lecture for the next one, so we can spend a good bit of time answering questions and reviewing if we need to before the test. All right, but please email me or text me. All right, guys. Bye. See y'all next Tuesday.